Hi and welcome everyone who has joined us here today to commemorate the legacy of our great mentor, Professor Sushil Kumar. Those who have joined us here today are witnessing the beginning of Professor Sushil Kumar Memorial Oration, which is a respectable tribute on his birthday anniversary by his students, colleagues, friends, and admirers. Joining us today, we have Dr. Manoj Prasad here, Professor and JC Bose National Fellow at the National Institute of Plant Genome Research, New Delhi, as our guest speaker. Hello, sir. We have Professor SPS Khanoja,
Hello.
Founder Chairman of Flora Fauna Science Foundation. Dr. Singh couldn't join us here today, but I guess he'll be joining us shortly. We have Dr. Neeraj Jain, General Secretary of the foundation. And with this, I welcome you all here. Now, without taking much time at this juncture, I request Professor SPS Khanoja to please initiate the session and share his views, thoughts on on our event today. Sir, please.
Namaskar. A very good evening to everyone. Uh, I would say that it's a great beginning of a great mentor's vision, which we want to translate through the orations and the expressions that would really make what he was dreaming in science. Uh, Unfortunately, we lost his uh, soul from this planet uh, last year due to COVID. But as you all, particularly those who have worked with him, those who are students, and those who are admirers, know that he's going to live because of the contributions he made and also the vision he passed on to us. I happen to be a direct student who has worked under him and learned how really discoveries are made. And I would say that I experienced a very different mentor to begin with. Uh, I would say that I was like an Eklavya for the Dronacharya, and then later I became more than Arjun for him because he always used to test his students that would they be able to do the things the way he wants. My association with him has been from 1980s to till he left us, but I can't believe that he actually is no more. So we thought that we will be keeping his legacy alive through involvement of all those who associated with him, and we would have the oration by eminent scientists who are going to be the leaders of future, and also we would have an award instituted in his name, which we will be giving to young scientists who are making something out of box, not a typical way of assessment, but we will be doing through a search committee. And this is an announcement that we wanted to make today. Today is very important because it is 14th of December in 1940 that he was born. I worked under him at IARI and also later part in CSIR, and had the privilege of learning directly of how to really be into aberrant part of the science where usually we try to ignore the results that we get, and that's what actually gives rise to the innovations. When we were looking at that who could be the orator of first of the series, uh, I observed that Manoj had published in Current Science an obituary, which actually was so deep, both sentimentally as well as scientifically, that I felt I don't have to search further. And I asked Manoj that, are you willing to be a part of this exercise? What we call this group is Dr. Sushil Kumar's school. He happily agreed. Well, I know Manoj from his start of the career at NIPGR. I was there in their research advisory council also, and have been observing the steady way he has been moving up on the ladder, and therefore it would be really wonderful to listen to him and the topic that he has picked up, feeding 10 billion, a genomic perspective. Last time I had heard him was in Bhubaneswar, and he was talking about the wonderful things that millets can do, and his papers and publications also are showing that the type of things that can be done with such crops, which are not traditional crops now, although millets had been the tradition of Indian food habits earlier. So, thinking back to nature and thinking that how complete nutrition can be provided is something that we are looking at from his side. It's not only the question of filling the bellies, but it is a question of making our metabolism such that we all are healthy. Just to apprise Manoj of our mission, our mission is uh, Potion Se Roshan, and Potion Se Roshan means that we want to empower the society with nutrition. And ultimately, the goal that we want to achieve is Prasanna Bharat, that India is happy, internally happy, and that can happen only when we are healthy, and also we have livelihood that really gives us the best lifestyle possible, so that we don't really fall sick. So therefore, with our keywords called health, uh, which is HE, and nutrition and agriculture, we name it HENA. And in that HENA, when this perspective of genomics adds value, we really would be moving towards the translation. And Professor Sushil Kumar always felt, whenever I interacted with him or our groups interacted with him, that science must reach the society, and in a way that it can really be adopted by the common man, whether it is a farmer or a villager or an urban inhabitant. And that's what we are trying to aim at. So we look forward to Dr. Manoj Prasad's oration today, and I'm sure that with his deep insight into the subject and also experience that he has in the field of genomics, we are going to be more learned today after this oration. I would now request Manika to formally introduce Professor Manoj Prasad.
So you have said so much about Professor Prasad already, such a reputed personality. But just brief introduction. Professor Manoj Prasad, born on 16 December 1970 in Balia, is working as professor and J.C. Bose National Fellow at National Institute of Plant Genome Research, New Delhi. He earned his PhD in botany from the prestigious University College of Science under Calcutta University. His research interest lies in plant molecular genetics, genomics, and stress biology. Dr. Prasad had published more than 150 research papers, reviews, and book chapters in highly reputed international journals, and is serving as associate editors in many renowned journals of high repute. He is an honorary adjunct professor at School of Life Sciences, University of Hyderabad, and a recipient of many prestigious awards and honors like Birbal Sahani Medal, Fellow of four national academies, that is INSA, NAS, IVS, and NASI, Allahabad. He has got Golden Peacock Award, J.C. Bose National Fellow, Tata Innovation Fellow, National Biosciences Award, Hiralal Chakrabarti Memorial Award, Humble's Fellowship, Young Scientist Award, and NAS Associateship. These are many awards and honors which he has received. And with this brief introduction, don't mind me, it's a very brief introduction of your personalities. So you'd like, we cannot just complete in few sentences. But with this, I request you to proceed with your lecture, please.
Yeah. Slide, the first slide. That
Yeah, sure. Just a minute. Just now you can see your slides and we can just, go ahead with the lectures.
yes. Yeah, yeah. So, good evening, everyone. It gives me a great pleasure to deliver the first Professor Sushil Kumar Memorial Oration organized by Flora and Fauna Science Foundation in association with ICR, National Institute of Plant Biotechnology, New Delhi. My foremost thanks are to the founder chairman, FFSF, and former director CSI CMAP, Dr. Suman P.S. Khanoja, and Dr. Anil Kumar Singh, ex-chief scientist CSR CMAP, for giving me this opportunity to deliver this talk amongst the distinguished audience. Professor Sushil Kumar was one of the pioneer researchers in plant sciences who had made a mark in the field through his research contribution in the field of genomics of crops and microbes, particularly his research on E. coli, lambda phage, and mutants of rhizobium. Also, Professor Kumar has been closely associated with NIPGR since its inception, and he has always found happiness in visiting us on almost all occasions till he left his heavenly abode. Personally, I had a very close association with him, and I always enjoyed working under his mentorship. He has been an inspiration to all, not only for to me, but to everyone who knew him and followed his work. The list of his students, colleagues, friends, and admirers is huge, who will always remember him for his service and contribution. At this moment, I profoundly remember Professor Sushil Kumar not only for his scientific achievement but also for his valuable contributions to the research community. I even had the pleasure of publishing a personal news article which I have shared with you people, recently published in Current Science. I request the audience to go through the said publication to know more about Professor Sushil Kumar. Today, my topic has relevance to the ongoing issue of hunger and malnutrition, as we all know that forthcoming challenges of how we are going to feed the 10 billion people by the end of year 2030. With this, I began my first Professor Sushil Kumar Memorial Oration on the topic Feeding 10 Billion and Genomics Perspective. For the convenience of the audience, I have made my topic very simplified, because if I give a scientific thing, that many people may not understand the subject of genomics. So that's why I have made oversimplified slides. So coming to the topic, what I'm going to talk about, how we can feed the 10 billion and how genomics or biotechnology can help us to do this. So, if you see that as of now, humans are the last one to evolve on this earth, but we are leading the game in terms of numbers. As of yesterday, this time our population count was 7.7 billion, and experts predict that it will reach to 10 billion by 2050. To this population, we have assigned three things as their basic needs, namely food, shelter, and clothes. We have been told that without these three needs, survival of a human is not easy. But if you ask that whether all the 7.7 billion gets the three basic needs, then the answer is no. 821 millions are still hungry. For every 10 seconds, a child dies from hunger-related causes. And 1 in 4 of the world's children suffer from malnutrition, which altogether means that getting food alone will not serve the purpose, but getting nutrition food will. If you see the global hunger index of 2019, and the Africas and South Asia, including India, still in the danger zone of hunger, irrespective of several steps has been taken, including the Green Revolution. The global hunger index scores, if you see from starting from 2000, 2005, 2010, 2019, at the regional level, you can see here South Asia and Africa south of the Sahara have highest hunger score in the world, with a number of 29.3 in South Asia and 28.4 in respectively in South Africa south of the Sahara. So this global report on food crisis for the year 2020 shows that 135 million people living in 55 different countries are facing acute food insecurity. This report was released before the incidence of COVID-19, and it is expected that the number would have been now increased due to several reasons, including lack of income and economic constraint among the vulnerable population. We are still far from a world without malnutrition, and this slide shows the statistics on the effect of food insecurity among the children. The Joint Malnutrition estimates that 144 million children under the age of 5 are stunted, 47 million are too thin, and 38 million are obese. If you see the percentage of stunted children, South Asia has more than 30% of stunted children. Among that amounts to 55.9 million children. In terms of wasted or too thin children, again we top with 14.3%, which amounts the 25.2 million children. So the situation is very grave actually in the present scenario. So realizing this grave situation to build the better world for people at all levels by 2013, United Nations formulated 17 sustainable goals, amongst which 3 are directly related to food. And this underlines the severity of the issue we are talking about. You can see here the 17 sustainable goal and these 3, no poverty, zero hunger, and good health and well-being. These are 3 directly related to food. When we come to food, I want to show important issue. The largest and most diverse botanical garden in Kew estimated that there are 11 lakhs of vascular plant. You can see here 11 lakhs of vascular plant. Among these, 7,000 were used as food by the early humans, which in due course of time shrunk to just 3 crops, rice, wheat, and maize. And these 3 crops cater more than 50% of the world food. In addition, there are 12 more crops or plant, and that altogether 5 animal species supplemented 75% of the world food. Now, you can understand that, that there is a great imbalance. So now the question is how we can feed the 7.7 billion with some 15 species. And this slide is where we can say that the high time to focus on mainstreaming the underutilized crops for food and nutritional security, and how this next generation genomics tool for trait improvement in this crop can be utilized. So coming to the first topic, the mainstreaming the underutilized crops for food and nutritional security, you can see that what is neglected. Neglected means that less research attention has been made, and what is underutilized, marginally cultivated and used. So this picture of millet shows that they are nutritionally are so important and high and nutritious as compared to any of the staple crop, rice and wheat, but still they are neglected and are underutilized. So if I come to know about what are the neglected underutilized crops, then we can talk about tubers, legumes, leaf vegetables, and minor millets. When we talk about the neglected underutilized crops, that means the neglected underutilized species are adapted to marginal environment. Neglected underutilized species are highly nutritious. They have potential for development as novel consumer products, and anyways are typically embedded within the local cultural tradition. So these are the 4 important points about the neglected underutilized species or crops. So for example is concerned, under tubers, we have taro, arrowroot, Indian root, ganola, purple yam, and air potato and elephant root. In terms of legumes, we have wing beans, jack bean, barbara groundnut, kidney beans, fava bean, lima bean, cowpea, and on many others beans, ajushi beans, and the velvet bean, rice bean, and horse gram. So these are the example of underutilized species. In terms of small or minor millet, we have finger millet, foxtail millet, prosum millet, little millet, barnyard millet, teff, kodo, fonio, guinea grass, brown steppe millet, and job steer. So if we count the state-wise estimate of millet production, although India is the second largest producer of millets after China, And when we talk about this area of cultivation, then Rajasthan and Maharashtra as at the top list. And in terms of yield per kg and hectare, Uttarakhand and Tamil Nadu are the first. So far as the genomic history and ecology of geographic spread of rice is concerned, that a recent study has showed that this rice has originated approximately 4,000 years ago, and the millets are already in existence before the rice has originated in a human civilization. So you can understand that these underutilized crop species, they are at one time they are mostly used by the human beings, but due to several regions, these crops have been replaced by major cereals like rice, wheat, and maize, and so on. But in terms of nutrition potential of millet, it has a high reach in protein, fat, as crude fiber, carbohydrate, energy, vitamins. And you can see here all the millets are there 3 to 5 times more superior in terms of all this composition per 100 gram of grain as compared to rice and wheat. When we talk about the micronutrient, again they are much superior as compared to rice and wheat. So millets are now slowly gaining the popularity. You can see that the media is now talking about it. There is one great example I can show you, that in Tamil Nadu, in Madurai, TNU, that Professor Vanni Rajan, he has developed one variety of Kodo millet, that is MDE1, which has replaced the production or replaced the rice production in terms of abundant millet, and that was cost in the market 110 rupees per kg. So you can see here that slowly millets are gaining that importance, and in southern part it was visible. And you can see here in COVID also the millets has been promoted, because due to this that are nutritionally much better than other staple crops. So keeping the importance of this millet, in 2018, the government of India through Gazette notification has termed all the millets as a nutri-cereals. And following this, the FAO, Food and Agriculture Organization of the United Nations, they declared the 2023 year is the International Year of Millet. So people, you can understand that everybody in the world and also in different parts, they are also recognizing that yes, millet is the crop one can do research and also come into picture. So now we ask the question, can millets address the hunger issue of ever-growing population? This integrated map shows the hunger hotspot spanning 55 countries with acute food insecurity population. This is the year 2022 20's statistics, and the small millet cultivation on those regions. And you can see here, this different color graph shows the acute food insecure population by 2020. Now, what is the roadmap? Recently, we published an opinion article on trends in plant science, where we advocated how we can take help of the small millet for enduring the food security amidst this pandemic. So we have to identify the millet species which is suitable for cultivation on the hunger hotspot. Then we have to optimize of the cultivation practice and training of farmers for millet cultivation. And then we have to do the implementation like large-scale production of millet species and optimization of downstream processing, and further the genetic improvement of millet, what we are doing right now, varieties using modern biotechnological breeding approach, and then the supply chain, that is the devising appropriate strategy for post-harvest processing and storage and supply chain. So this is the roadmap what can be utilized for betterment of this underutilized crop species. This is just millet is one of the example of this. Now the big threat is agriculture is on its way, because as I said in my title, that 10 billion people to feed by 2030, then and the recession is there, and then this COVID problem, and then there are several biotic and abiotic stress and natural calamities. So these are the severe problems that in keeping in mind, we have to think how we can circumvent this thing. Coming to the next part of my talk, how we can use this next generation genomics tool for trait improvement in this underutilized crop species. Now we know that the breeding is very slow because till 10 years back, all the variety, whatever we have developed till date, they are all through this normal breeding process. A classical example was IR development of IR8, the semi-dwarf with good yield. By crossing between Peta Indonesian tall variety with low yield with DG gene of the Chinese variety with dwarf with normal yield, and we have gotten semi-dwarf with good yield IR8. And this is the history how this IR8 has been developed in International Crisis Institute in Manila. So breeding is still slow because this slide shows that it took almost 10 years to release one variety through different breeding approach like forward breeding or the backcross breeding or any other approach. So breeding is still slow, and it will take much longer time. So what is the need to expedite the crop improvement program? So we need some alternative technology. And to do this, again, the climate is changing very fast. You can see here that India has 2% world land, 4% of fresh water, but 16% of the world population and 10% of the cattle. So, and also scientists have found that the inland areas are far in risk rather than this coastal area or hilly areas, because the heat is one of the severe problem and in the inland areas, and you can see here heat is severe problem as compared to the coastal or the hilly areas, because this temperature and other stuff they can maintain by the hilly areas and the coastal areas, but not in the inland areas. So crop failure due to climate change is very fast. So what is the problem? There is no rain, late monsoon, heavy monsoon, pest, insect, and disease, and other natural calamities. So these are the causes of crop failure. So recently we have seen that the pest attack were increasing. You can sorry, this video is, I think, is not working. There is one video I have. So anyway, so you can see here that the pest attacks were increasing. So this locust attack recently, that last year, that it lost all the fields, whenever it was attacked. So this is a natural calamity. You cannot do anything in this such kind of situation. Again, FAO has estimated that that the drought is one of the major problem. It causes 83% of this crop loss, followed by volcano eruption, storm, food, and flood, and tsunami and earthquake. So drought is a severe problem and the current climate change condition. So this national report that shows that more than 42,000 farmers has been and daily wages committed suicide in 2019. But this is before this pandemic arise. So you can see here, this is a statewide data. So the question is, when all the farmers will gone, then it will be our turn. So we have to be cautious, and we have to find out some solution how to tackle this climate change. And this will continue. By 2050, crop yield will decrease by 20% if we don't address the climate change right now. And this is the estimate by the Food and Agriculture Organization of United Nations. So, can biotechnology rescue agriculture? So the answer is, I will show you in some with great example. So we know that there are different stresses, and then to circumvent these stresses, we have biotechnological intervention. So in terms of different stresses, we have drought, flood, cold, heat, salinity, radiation, oxidative stress, and pathogen. In terms of biotechnological intervention, we have several omics technology like genomics, transcriptomics, proteomics, metabolomics, epigenomics, ionomics, and phenomics. So in layman language, if I want to say, how could we use this biotechnology? And so a simple answer is this: if we compare the sequence of high yielding plant with a low yielding plant, we will found that there is some gene or locus that has some additional sequence in the high yielding plant. You can see here there are 5 pods here, there are only 2 pods. So in the high yielding plant, some portion of a gene is extra, but that is absent in the low yielding plant. So can we transfer this gene from high yielding plant to the other one, which is a better yielding plant? So the answer is yes, through genetic engineering. And now the question is how the DNA sequencing has revolutionized the crop improvement program. I can show you that. So once this genome sequencing thing has been, NGS platform has came in the year 2010, and lot of genome sequence is available for many crops. Now it is more than 100 crops is available. So we generate some data like ATGC format. This I called as a noise. So we have to make sense this noise, and that there is several computer programs that can sense to find out the genes which encodes the protein. You can see here the genes, and it is depicted in the red color, and the regulatory motifs that controls the expression of the particular gene. You can see here in the different red colors. Now, as I showed in my previous slide, that we have identified the gene of our interest, which is responsible for the higher yield. Then how do you transfer this? For that, first we have to identify the gene or the locus. Then we have to clone this in a vector, and then we have to transform it to through the explant to the transgenic, to raise the transgenic plant. So by this way, we can generate a higher yielding plant from the candidate genes. And to do this, we have several methods. Like one of the method is Agrobacterium mediated gene transfer. This the gene can be from any source. It can be bacteria, it can be human, it can be fungus, for any source. We have to clone this gene in a vector in terms of plasmid, and then this plasmid, with the help of Agrobacterium, we can introduce into the plant, low yielding plant or the susceptible plant, so that the gene can introduce, and then the transgenic plant have high yielding or this resistance. There is some great example of this BT revolution. You know that BT has causes severe damage to the crop plants, and how to circumvent this. And for that, the scientists have identified that yes, Bacillus thuringiensis kills the insect. So this is the bacteria, Bacillus thuringiensis. This is known as BT, and in this bacteria there is a toxin, that BT toxin, that is crystalline structure, protein known as. When the insect eat this BT crystal protein, it dissolve in the gut of the insect, which is very high pH, alkaline pH, and this toxin attack the gut cell of the insect and makes spores, and that's why the insect die because of starvation. So this is the way that the gene from bacteria was introduced in plant when the insect attack the plant, eat the leaf of that BT gene containing leaf, and then they have died. So how this has been done? That, as I said, that this the BT toxin, the crystal protein, this is the structure of the protein, and then this was cloned in the vector, and then through ectobacterium transformation, it can be introduced in the plant, and then this transgenic plant containing the BT gene. So there is some more example, like you can see here the non-BT brinjal and there's a BT brinjal. So the non-BT cotton and the BT cotton, and this is the non-BT and the BT corns. So you can see here the difference of this. It's such simple example of introducing one bacterial gene to a plant system. Coming to our own example, how I have shown my previous slide from a gene from a bacteria, how it can improve the plant in terms of disease resistance or yield. Now coming to the plant-to-plant gene transfer. So the genome of foxtail millet has been sequenced in the year 2012 by the Chinese group and the American group simultaneously. So with this genomic information, we have made a comparative survey, and then we compare the foxtail millet genome with this sorghum and maize, And we identified that 586 genes were functionally annotated as a response to water. And further re-annotation, we have hits of protein, aquaporins, MIP transcription factor, Werke transcription factor, and ADP ribosylation factor. So these are the different candidate genes we can work on. And the classical example, we have found that one small hits of protein in foxtail millet, which is basically is a C4 panicoid specific, because this gene is not present in any C3 grasses. So through our effort, we have identified from the expression data that HSP27, the small hits of protein, is specific to C4 grass, panicoid grass, and not present in any of the C3 grasses. So we isolate this gene, clone this gene, HSP27 of foxtail millet, and we introduce it into the rice plant. And we develop some transgenic rice lines which have better improved agronomic traits in terms of plant height, tiller number, panicle number, size, and number of seed per plant panicle. So better yield in terms of at 50 degrees centigrade. So one plant to plant gene transfer I have shown. Now how this gene is working, it's a very simplified way. When this is the normal gene, and then it produces the normal protein. When in extreme heat condition, above 40 or 45, the normal protein denatures, and that's why the plant dies with no function. But when we introduce this HSP27 gene in the plant, rice plant, and this HSP27 binds to the some other proteins to prevent their degradation during extreme heat, and that's the reason why the rice plant is surviving with HSP27 of foxtail millet gene at 50 degrees centigrade. And this we have taken Indian patent. And now I want to show you some important thing that why we characterize gene families, because as you know that I have talked about HSP27, I have talked about there were several transcription factors, MIP transcription, Werke transcription factor, but you know that we cannot work out more than 100 or 120 candidate genes. We have to identify the candidate genes for our overexpression study. And to do this, we have developed one pipeline. Like we identify the gene family members for particular transcription factor. And for example, here I can give you example that we have identified the small hits of protein in foxtail millet, 113 hits of protein we identified. We performed the gene structure, chromosome location and duplication and the divergence study. We have studied the domain architecture, phylogeny and physiochemical properties of all these 113 HSPs. And then we have done the upstream analysis like promoter analysis and gene ontology. And then we have done 113 expression profiling in response to abiotic stresses. And then we have done the overexpression in each system, in one heterologous system, to get one first-hand data within 10 to 15 days time period. So by this way, from 113 HSP gene, we have found out the HSP27, which is differentially expressed in all the 4 different abiotic stresses like heat, droughts, salinity, and cold. So other 112, they are not all are differentially expressed in all the 4 abiotic stress condition. And this way we have identified 124 C2H2 type zinc finger transcription factor in foxtail millet, 110 Werke transcription factor. So, but as I said that we cannot work with 110, 124. We have to find out the candidate gene 1 or 2, and that is the way I have shown you that this pipeline has very important to identify the candidate gene for overexpression or gene knockout or knockdown. So there are different biotechnical approaches are now available to fine-tune the genes. The first one is the overexpression. The overexpression means overexpression of a target protein. As I showed you, like BT, and I have shown you the HSP27 for our case. There are 2 other approaches. One is RNAi, where we silence the target protein, and CRISPR-Cas9, where we mutate the target site. So this is a knockdown and this is a knockout. So another example was overexpression of a candidate gene. This is a Citari italica autophagy gene, which confers tolerance to nitrogen starvation in rice. Like similarly, I have shown you the HSP27 story. The Chinese, they have shown the one foxtail millet autophagy gene, ATG8A. When they overexpress this gene in rice, it can tolerate in a very low nitrogen content, starvation condition. There is another great example of overexpression by foxtail millet gene ASR4, which is abscisic acid stress and ripening induced gene, and they overexpress in rice, which confer tolerance to salt and drought stress. So this panel shows the drought, and this is the salinity. These two lines are the overexpression line, and these two lines in the RNAi. So this is overexpression, and this is the knockdown. And this is middle panel, you can show you that is the wild type. So when this is the normal control condition in both the cases, drought and salinity. And when in the drought, you can treatment for 10 days, and in drought for 13 days, and recovery in 5 days. You can see here the overexpression of this SIAR4, the plants are healthy. And when the same gene was knocked down, the plant dies. So both overexpression and RNAi knockdown shows, scientists show that this gene can work both for drought and salinity stress. Now, as I said you that knockdown and knockout, so there is an example. I have shown you RNAi, and now I am going to show you some VIX example and knockout and the CRISPR. So this is an example of RNAi. This is again a single BASD gene of Citeria viridis. Citeria viridis is actually the wild progenitor of the cultivated foxtail millet. So this is wild. And this BASD is a gene of beet, AHCT, HCVT, and DAT. So the combination of 4 single BASD gene in Citeria viridis causes large stable decrease in cell wall ferrurization and increased biomass digestibility. So this is one great example for RNAi. So what is VIX? VIX is basically virus-induced gene silencing. It's the fooling a plant gene to degrade its own RNA. So what we did, we want to silence that plant gene, which is we want to know the function, or we want to get rid of this. So to want to know the function of a plant gene, what we did, we cloned this plant gene in a virus, VIX vector, virus-induced gene silencing vector. So this recombinant viral RNA, it was cleaved by the RISC and Agio complex. So basically, the plant gene was cloned in a virus vector. So it embedded in the recombinant viral RNA. It produces the secondary structure, and then dicer cleaved it into the siRNA, and siRNA cuts this mRNA into different fragments. So when it cuts this viral RNA, it also cuts the plant RNA. So by this way, we can silence the plant gene by taking advantage of the viral vector. Another example for the knockout is CRISPR. It's a very new technology, and a lot of work has been done. So CRISPR is a combination of this Cas9 protein, that is the Caesar, and the guide RNA. So the guide RNA here, this is the guide RNA, and this guide RNA sequence matches with the target gene in the genome. So this is the target gene, and this guide RNA helps the matching in the target gene where we want to do the knockout. So it consists of 2 components: a single guide RNA, and it is customized based on this target gene, and the Cas9 protein. This is as an endonuclease, and that cuts the cleavage on in the target site. And then the Cas9 assembly bind to the target gene, and it cuts the nucleus of the Cas9 cuts the target gene, and by this way, this knockout this thing. And then the DNA repairs happen that actually is error prone. So there's great example of CRISPR-Cas9 to prevent pre-harvest priority tolerance, a major issue on wheat. So if you see that sometimes in the field when rain is there, actually uncalled rain is there, some of the spikes they sprout, and it causes the yield loss and the quality of the grain. So, and you know that the bread wheat has three genome, A, B, and D. So for CRISPR-Cas9 or genome editing, you have to edit all the three gene to make it successful. So scientists have done this, that they have identified one gene, that is QSD1. This encodes an alanine aminotransferase family members. And this alanine aminotransferase gene member, they have done this triple knockout mutant, and you can see here the TCCCTGAC. So they have introduced this ATT in all the A genome, B genome, and D genome, and this triple knockout mutant makes this dormancy period, increase the dormancy period longer, so that there is no sprouting. So through CRISPR technology, scientists has produced a longer seed dormancy period in the wheat plant, so that if rain comes or if there is anything comes and it cannot sprout. Another great example is the de novo domestication. So this is a wild tomato. This is known as Solanum pimpinifolium. So this wild has a property like indeterminate growth. You can see here pea-sized fruit, very small fruit, and stress, but all the wilds are stress resistance. So through CRISPR technology, using CRISPR by editing six genes, genes related to plant growth habit, fruit shape, fruit size, 2 genes, fruit number, and nutritional quality, lycopene beta cyclase. These 6 gene scientists have mutated by the CRISPR-Cas9 technology, and they have made this Solanum pimpinifolium wild to domesticated Solanum pimpinifolium with determinate growth, large and more fruit, 500% of high lycopene, and stress resistant. So look, the beauty of this technology, that by editing 6 genes, you make a wild plant to a domesticated cultivated one. So this is the wild, and this is the domesticated the wild plant. So increasing 500% of lycopene. So in conclusion, I can say that molecular breeding and genetic manipulation have emerged as the 2 most potent technology for crop improvement. Advances in NGS technology have enabled the incorporation of genomics with various disciplines of crop breeding. Functional and comparative genomics have provided the platform for gene discovery and their functional characterization. The key gene or genes regulating a molecular pathway are being genetically engineered or edited to develop phenotypically improved crop lines. Collaborative and concerted research is required to circumvent these global laws. And I must show this slide because a mentor is nothing without the achievement of the past and present students. So I can say here that I am very proud of my students, because whatever awards and achievement I have today, that is all because of these brilliant guys actually. So Charu did a PhD with me. She characterized the SI dehydration responsive element binding protein, which conferring drought resistance in foxton millet. She got all the awards available at below the age of 35, and she initially got selected as a scientist at NVRA Lucknow. And Namisha, she characterized the microRNA, which is responsible for providing tolerance against the tomato leaf colony virus. Recently, we have published a paper in PNS, and she is the first author. She got the INSA Young Scientist Medal in 2019, and now currently she is working as a Sir Women Excellence Awardee. Muthu also, like Charu, he got everything below the age of 35, and he got assistant professor position at University of Hyderabad in plant science department. He performed all the bioinformatics and genomics resources development in foxton millet during his PhD. Swati characterized the NAC transcription factor in which is responsible for salinity tolerance in foxton millet. She got the Marie Curie Fellowship. Pranav characterized these ubiquitin proteasomal pathway genes, SLRPT4, providing tolerance to tomato against the tomato leaf colony virus. He also got the Marie Curie Fellowship. Ashish currently submitted his thesis and got his awards. Now I should write him doctor. He has done excellent work, and he for the first time he started work in autophagy, and he developed some good markers for this autophagy and for providing tolerance against this tomato leaf carnivore virus. And he also characterized ATG8F, how this ATG8F gene, autophagy gene, has provided tolerance against tomato leaf carnivore virus. He also got the Newton Bhava Fellowship for a brief period. He was at UK at Ari Sundaram's lab in Durham University. Others characterize the WD40 protein, which confers the hepatic stress tolerance in foxtail millet. He got the Brave Scholarship in 2015. So all these students, they are, whatever they do, it will face them some good awards and fellowship and a good postdoc. And a few of them also absorb in regular position. So thank you very much. I must acknowledge the Department of Biotechnology and Ministry of Science and Technology and NIPGR for providing funding for this work. And this is the dynamic group you can see here. Ladies as the first, and we followed them, and we thought that they are our driving force, and so they lead us in all aspects. Thank you very much. And I hope that I try to convince you that underutilized crop species is the need of the hour, and we must work hard to answer all these questions. What people has done for all the major crops, rice and wheat and maize, but for underutilized crop, very less people are working, and very less group is putting effort. So we must concentrate on this underutilized crop species so that in coming years we can address the global food crisis, and so that we can feed all the hungry people. Thank you very much.
Thank you so much, sir, for such an intriguing talk. I guess I can now open the talk to for a panel discussion kind of thing for some concluding remarks by Professor Khanoja. So please, can you please unmute your mic, sir? Yeah.
Thank you, Manoj. It was a wonderful talk. Uh, we did look forward to such a knowledge enriched message that you are going to deliver through this oration. I'm sure that particularly the students and scholars who are making their career ahead in biotechnology and genomics research would find it as one of the eye openers in terms of the opportunities ahead. If I have to conclude or summarize what Dr. Manoj Prasad talked about, it goes into two directions. One of the directions is that goes into crops, and he did talk about underutilized crops. I look at it from another angle, and that is what we call as ancient grains. Now, ancient grains are in one way defined by people with the age or the knowledge that when they were used first of all, but then there are checks and balances on that. But just to share with everyone that the most ancient grains that human civilization has used have been pulses, and as we know that pulses are the main source of protein, and deficiency of protein is a major problem leading to stunting and many other causes of metabolic disorders. And on the other hand, the fiber and the lipids are important in combination with the proteins, and also micronutrients and vitamins. So that is where millets start getting uh, a top edge over many other crops. We were using them, so they were ancient grains. Today we are looking at them as future crops. So a journey from ancient to future is going to happen. And when we combine the traditional genetic tools and the modern genetic tools, a combination is going to give us many answers. So that is how these are going to be important. There are some other type of definitions given to such ancient grains like pseudo cereals, which is again very very important. And in a way, if we look at all these crops, whether it is millets or ancient grains, what we call, or pseudo cereals, one of the common thing is that they require less of water, and they can really adapt to the changing climate. And therefore, in future, even if we don't want, these crops only are going to give us the genes and the genomes that we can really borrow part of that and then bring into any crop that you are using as food crops. And the other message that he gave is in the genomic perspective itself, and that is, what is desirable, overexpress it. What is undesirable, silence it. And what is not behaving, modify it or edit it. So these are the three major ways of manipulating the genomes as well as the genes. And obviously, when we are trying to do that, one of the examples that he gave of a wild tomato towards domestication also indicates to us that it's not only borrowing of the genes, particularly qualitative genes, but also the wild germplasm that exists in nature, and we have to go back into the wild and look for that germplasm. Many answers that we are looking at in terms of manipulating the genomes or bringing in the new traits, whether from bacteria, fungi, plants, or animals, could also be present in the plant by itself for so many years. Again, it's the ancient resources only. So that re-looking at the gene bank of the germplasm would be very, very important. And comparing it with the recent situation that has happened uh, with corona, I would say that the corona virus is so smart that it opted for the third strategy that Dr. Manoj was talking about, that is gene editing. And it is what we are looking at today at Omicron, as a cluster of mutations have happened. I believe that it could be not just the mutations, but it is the general recombination that has happened in patients or in the carriers who were having more than one pathogens in them, or more than one viral variants in them, or maybe both of them did not look infectious, but when they recombined, the clusters of mutations that we are seeing in Omicron has happened. And this is the basic genetics that happens in viruses through general recombination. So that also tells that how we could be thinking not necessarily with scissors and paste, that means cloning, but also through a breeding approach, which he touched upon as molecular breeding. That is going to be the future. So I must congratulate Dr. Manoj Prasad for being with us and giving such a knowledge-rich oration. And we look forward to continuation of his association with Flora Fauna Science Foundation, and also the activities that we do to take legacy of Professor Sushil Kumar ahead. So thank you, Manoj, for such a wonderful treat that you have given to the whole society.
Thank you, sir. Thank you. Thank you, sir.
Over to Dr. Neeraj for the vote of thanks.
Thank you, Dr. Manika. Good evening, everyone. Now it's time to place on record the vote of thanks, the responsibility that I have been given. First and foremost, I would like to place on record my sincere thanks to Dr. Manoj Prasad, Professor and J.C. Bose Fellow at NIPGR, for graciously accepting the invitation to deliver this first oration of the series. It was a very wonderful and informative lecture, sir. Thank you so much for joining us. With this lecture, the series has taken a great start and set the bar very high for future orations. Now I would like to thank Professor S.P.S. Khanoja, Founder President, Flora Fauna Science Foundation and ex-director, CMAP, the genius behind conceptualization of the oration series to commemorate the legacy of great mentor Professor Sushil Kumar. Under his guidance, a group has been formulated as Professor Sushil Kumar's school, as Sir was telling in the beginning, and an award has also been instituted in his memory. Further, I would like to thank all the people who have been directly or indirectly instrumental in making this oration possible. They have been students or interacted with Professor Sushil Kumar at some point or other in their career. Placing on record a few names: Dr. Ajit Kumar Sashini, presently Director NIPV; Dr. M.P. Darokar, Senior Principal Scientist, Technology Management Directorate at New Delhi. I would also like to thank Dr. A.K. Singh especially, who is Vice President, Flora Fauna Science Foundation and former scientist at CMAP, who has been very instrumental in making this oration series possible. He has been working at different levels to make this oration possible. And last but not the least, I would like to sincerely thank Dr. Manika, who organized the oration on the online platform and linked to all other social media platform where it is streaming live, and also it will be available for all our audience for later references also. Thank you. Over to you, Dr. Manika.
Thank you so much, Dr. Neeraj. Thank you so much. Thank you, sir, for joining us here. So you're on mute as
Yeah.
first.
So I would also like to uh, announce that as a gesture, for this oration, we would like to confer honorary membership of Flora Fauna Science Foundation to Professor Manoj Prasad. Please accept it.
Thank you, sir. Thank you, sir. Accept it.
Thank you, all of
Welcome.
you, for joining us here today. Thank